Okay, Summer, make sure you grab all these latecomers. So I want to welcome everyone to the Judith Tam University of Michigan ALK Positive Initiative ALKtALK. It's a lot of words. And I'm going to we're going to skip over the polling questions today because we have a lot to cover. And I want to introduce Kirk Smith, who is president of ALK Positive. Go ahead, Kirk.
Hey, Mark. Thanks everybody, and welcome to tonight's ALK Really just wanted to let you guys know if any of you are considering or still considering coming to the ALK Summit this summer in July in Philadelphia, that we are quickly running out of in-person spaces. Right now we are close to 210 that have registered. We're taking a max of 250 in person. So if any of y'all are still thinking about it, I would encourage you to go ahead and make a decision. And because we don't want you to get left out if you're really interested in showing up. But also, just as a reminder, it will also be virtual, and we don't have a limit on the numbers for virtual. And last year, I think we had close to maybe a little over close to 1,000 that were virtual. So you're welcome to do that. But we know a lot of people are excited about coming to Philadelphia. Obviously, we've almost filled up in a week's time. So just want to give y'all a heads up about that. So with that, welcome. And I will pass it along, I think, over to Jeff. Is that correct, guys?
Yeah. Good evening, good afternoon, wherever you are. Good morning if you're in Europe, I suppose. And good tomorrow if you're in Asia. It's my great pleasure to introduce Dr. Sophia Moriver and Dr. Angel Chin of the University of Michigan's ALK Lung Cancer Initiative. And with that, I'll turn it over to them and let them walk through their agenda, and then we'll open it up later for Q&A.
Great.
Angel?
Yep. Thanks, Jeff. Hi, everyone. I'm Angel Chin. I'm an assistant professor at the University of Michigan. And I think we thought we would just start off tonight with a quick set of PowerPoints about an upcoming clinical trial that we're working on at University of Michigan through the generosity of ALK Positive, who is helping to fund this study. So let me see if I can share my screen. All right. Does everybody see that?
Yep.
Okay. Let me put that in.
Yes.
All right. So I want to talk a little bit about our study, which is called Gilteritinib for lorlatinib resistant ALK non-small cell lung cancer. I really love this graphic. It was just recently published in a review in Nature Cancer from Dr. Schneider, Dr. Jessica Lin, Dr. Alice Shaw, and they really highlighted the mechanisms of ALK resistance. I'm sorry, my cat is trying to get in on this conversation. And they really break it down into on-target resistance mechanisms as well as off-target resistance mechanisms. So on your left, we see on-target resistance mechanisms. And by that, we mean really mutations within ALK itself. So we see that there can be single ALK mutations, and this usually happens when you've been exposed to first or second generation ALK inhibitors. And then you can have compound mutations, which can happen with exposure to third generation ALK inhibitors. If we look on the right, off-target resistance mechanisms can be divided into what we call activation of bypass pathways or lineage transformation. So when we talk about bypass pathways, what we mean is that the cancer is no longer reliant on ALK. There are other pathways that are activated, and some of these are listed here: EGFR, HER2, MET is actually quite common. And so these pathways can become activated, and so that ALK inhibition alone is no longer effective. And then when we talk about lineage transformation, what we mean is that just like in EGFR non-small cell lung cancer, ALK non-small cell lung cancer has been shown to transform from adenocarcinoma to squamous cell or even small cell carcinoma, also making it more difficult to treat. This is another paper that is coming out of Dr. Aaron Hada's lab from Massachusetts General, and they really looked at what happens to patients who've progressed on lorlatinib, which I think is the most clinically concerning population right now, since that is our current and only available third-generation ALK inhibitor. So that took 48 biopsies from 47 different patients who progressed on lorlatinib, and that's seen on the pie graph on the left. And they showed about half of these patients no longer had a detectable ALK mutation, suggesting that perhaps they had this ALK-independent pathway that was driving the resistance. We saw about 20% of these patients have a single ALK mutation, and then about 30% of these patients developed a compound ALK mutation. If you look in the middle, the researchers also looked at what is the sensitivity of these compound mutations to these different currently approved ALK inhibitors. And for me, it's a simple graphic, red is bad. And so if you look at these different ALK compound mutations, you see that they generally are resistant to pretty much all of the currently approved ALK inhibitors. And then if you look on the right, the researchers also tried to study how these mutations came about. So some of these mutations were detected even pre-lorlatinib, and that's marked with a little white star. And then some of these mutations develop post-lorlatinib, and that's just in the solid blue. We also talked a little bit about ALK-independent resistance mechanisms, and as you can see here, there are a slew of different bypass mechanisms that have been either reported in simply case reports or studied a little bit more commonly that can become activated when patients are exposed to ALK inhibitors that then help to drive resistance. Some of these we do have targeted therapies for. So, MET exon 14 skipping mutations, we have medications approved, RET, EGFR, HER2. But there are also other pathways like SHP2, AXL, et cetera, that we don't currently have any approved targeted drugs. So for me, what would be the features of an ideal post-lorlatinib TKI? So in the ideal world, I think that this drug should have activity against a single ALK mutation. There's some ALK mutations that even as a single mutation convey resistance to lorlatinib. Of course, we would like to see activity against compound ALK mutations. And the G1202R and the I117N mutations are the most common partners for these compound mutations. And in the most ideal of worlds, we'd also like to see activity against ALK-independent pathways. So with that in mind, here is the reason why I am interested in a drug called Gilteritinib. Now, it is already approved as a TKI. It's already approved by the FDA, but it's for the treatment of something called a FLT3 mutated acute myeloid leukemia. So a very different disease state than ALK non-small cell lung cancer. The reason I'm very interested in this drug, though, is that not only does Gilteritinib have activity against FLT3, it also has activity against all these other kinase domains, including ALK and non-ALK pathways that can be involved in ALK resistance. And here on the bottom, you'll see that the researchers who published this study out of Japan, they actually took cell lines that had either single ALK mutations on the left, compound ALK mutations in the middle, and some very resistant compound ALK mutations on the right. And they looked at, did Gilteritinib, which is the little star, have activity against these mutations? And seemingly it does, at least in these cell lines, that there's activity of Gilteritinib against these mutations, and perhaps at a lower dose than what you expect with lorlatinib and alectinib. So here is some patient data. So this is from a pleural effusion of a patient who had ALK positive non-small cell lung cancer, who was able to send us their specimen, and we did some drug testing on their specimen. This patient had progression on alectinib and lorlatinib, and so you can here see that it is in red on the bottom, but our drug testing platform would actually predict that the cells, at least, are sensitive to Gilteritinib. And so here is our proposed study. It's a phase 1 study of Gilteritinib in non-small cell lung cancer that's ALK positive. This protocol is currently in the works, so we are working on finalizing the protocol, making sure that the pharmaceutical company, the makers of Gilteritinib, are completely on board with the protocol, and then of course moving through the approval process with the FDA, IRB, etc. But really, my goal in this protocol is to be able to enroll patients who've progressed on any number of ALK TKIs, crizotinib, ceritinib, brigatinib, alectinib, and lorlatinib. Then, of course, recognizing that some patients have already gone to receive platinum doublet chemotherapies or even other anti-neoplastic therapies, including clinical trials. So we really want to be inclusive in this study. And with that is it from me.
Great. Sophia, you want to take over?
Sure. Yes. Good evening. Thank you so much to the ALK leadership, ALKpositive.org leadership for inviting me. It's a pleasure to meet your new CEO, Kirk, and hopefully get to meet a lot of you here or in lung meetings. And as always, we love to hear from you. So I will tell you about the initiative in general, and I think there'll be opportunities to ask a lot of questions and so on. Okay. It doesn't seem to want to. Okay. I don't need to record anything, so it doesn't seem to let me share the screen, but maybe now it will work. Okay. Is everybody can see the screens? Okay, yeah.
Yes.
Right. Okay, thank you. Thank you so much. So I want to tell you about the University of Michigan Judith Tam ALK Lung Cancer Research Initiative. I know it's a handful, but every word matters in that title, and it is through the generosity of Judith Tam that we've been able to work nonstop this last year in doing the best we can to really untangle a lot of very pressing questions we need to understand about ALK lung cancer, and that will put us on a track for a number of advances, both in how to treat patients at this time, in the present and in the near future, plus how to possibly even prevent the disease, how to prevent recurrence, how to prevent metastasis. A lot of aspects of the disease are really not well understood. And we have had the privilege of being able to work nonstop this past year on this topic. So Angel Chen, Dr. Chen is the clinical lead and most important, really, the most important person really in this initiative, because it will be her stewardship that will bring the science to the patients. And this is an initiative for patients. We want to do a lot of basic science that it is still geared to understanding pressing problems going on in patients right now. Stacy Fry is our program manager. Many of you have met her or talked to her. Nikki Pfefferly is personal administrator for my program in general. And then we have a senior accountant to make sure I don't overspend all the accounts. Let me see, how can I advance the slides? So as I said, this is a program to have utility for patients now in the immediate or near future. We believe that that's a goal that is seldom contemplated in the science of oncology. Even though we talk about precision oncology, we typically don't talk about precision oncology for you or you or you right now. So we have specific programs, specific projects described in the agreement with Judith Tam, and we are adhering to those goals. We want to create science that will help all patients, no matter where they live, no matter what their income is, what the color of their skin, what access did they have in the past. We would like to equalize all previous injustices of access. And when it comes to ALK, non-small cell lung cancer, all the highly technological advances that we and others are crafting, it's a major, major goal to have equity. And that's one of the reasons it's such an honor for us, for Dr. Chin and myself, to be speaking to the patients directly. Because ameliorating disparities is the most important part, because it turns out that there is quite a bit of understanding that highly sophisticated technologies have the very much an immediate effect of exacerbating disparities. And so we are going so far as to partner with the School of Public Policy, the Ford School of Public Policy at the University of Michigan, which is world-renowned. And we are going to have a postdoctoral fellow dedicated to making sure that everything we do, we are thinking about equity for patients. As we develop the technology, the usual way to do it is to develop the technology and then find that there is inequitable access, and then you try to fix it. We'd like to not break it to begin with. And so that is the goal of that effort, and we're just starting with that in our second year. We are training the next generation of ALK lung cancer researchers. There is a lot to do in this disease, and it is our deep obligation to make this effort sustainable, both through funding, additional funding, and through the training of researchers. And the key, of course, of bringing science to the patient is going to be collaborative clinical trials that have strong correlative science based on the discoveries. So in the immediate future, present, and future, we are developing and validating real-time drug testing platforms for ALK non-small cell lung cancer to enable precision decisions longitudinally. So if there is progression, what to treat with next, do you treat with monotherapy? Are there clinical trials for dual therapies? Are there scheduling differences, etc.? A number of different questions can be answered through our drug testing platform. Unfortunately, this disease remains quite mysterious. There is at least one major paper and several others that hint at the fact that the ALK fusion may occur even years to decades before the disease manifests itself. So that tells us that there is a window of opportunity for understanding and perhaps even preventing, once the understanding is there, how does it all happen to begin with. So we have a whole project devoted to understanding in depth the mechanisms of how the tumor arises, and then how it progresses under certain different types of influences. Now, you just saw a beautiful paper with 40-some patients. It turns out that if you add up all those mechanisms of resistance that Dr. Chin quoted from the paper, and there are, of course, others. Now, if you have only 45 patients, you almost have a different mechanism for each patient, if you take into account the mutations as well. So that paper only reinforces our determination to really understand each patient. And this is a challenge. And that's why we are trying very hard to test as many patients as possible, because no two patients are the same at the molecular or drug sensitivity level. We want to understand how the cancer begins in the first place. this is an unusual situation. This is a fusion where the kinase ALK takes on a function that, sure, it's related to the kinase, but there are other things going on in the cell with this fusion protein floating around the cytoplasm where it normally wouldn't be. And we know that because we know that different variants of different sizes have different sensitivities to drugs. So we know that the actual structure of this fusion protein matters. It's not just the kinase that's causing the trouble that gives rise to the cancer. There are other consequences. And so in the process of understanding that, I think we will come up with very important new discoveries for patients. We have core members in our team. It's a very large team, probably not everyone listed here. We have postdoctoral fellows, and we have a large staff of technical PhD prepared, master's prepared, recent graduates who help us manage the sample collection, manage the databases, etc. Okay, I hope I'm still, okay. So this is the structure of the initiative. A lot of people wanted to see the whole initiative. So we have, as I said, the lead investigator, is Dr. Chin, and I am the overall lead. We have, of course, an IRB approved protocol. We have animal protocols. We have a bioinformatics core and biostatistics core that serve all the projects. The program manager and the advisory board and members of ALKpositive.org are in our advisory board, and very important members, and scientific expertise broadly to cover all the projects. Project 1 is the project of deep understanding at the molecular and drug sensitivity level of patient material. So it's at the heart of the initiative of serving the patients today, right now. Project 2, led by Peggy Sue and Jason Spence, is a fundamental research program to understand how ALK cancer arises in the first place, how it evolves under the pressure of therapy, and how it occasionally transforms, what other cells are implicated, and so on in its progression. Project 3, a project I also participate in, is led by Matt Zollner, a medicinal chemist, where he and his team are developing truly new drugs. And they are taking several strategies of attack. Matt is a long-standing kinase expert. So kinase inhibitors are his expertise. And so he is developing new ALK kinase inhibitors. And at the same time, we are developing drugs that degrade ALK. That is, they accelerate the disappearance of ALK from the cell. And those are called PROTACs. So they encourage, they have their whole hands. One side holds hand with ALK, the other hand of the drug has two hands, holds hand with the machinery of the cell that degrades the proteins. And so it brings the degrading machinery to ALK and degrades it. And we're very excited about the new drugs that we have in the pipeline already. Matt synthesized 400 new compounds, never before synthesized in the world, so original compounds. And of those, there are 3 or 4 very nice compounds that achieve very good degradation of ALK within 24 hours. So we are trying to advance those and understand whether they will be good drugs, whether they could be taken orally, etc. All the things that one has to do to develop a drug. There are 2 developing projects that I think will be of great interest to patients. One is understanding why is ALK non-small cell lung cancer so resistant to immune therapy. So trying to understand what does the immune microenvironment of specific patients, how does it contribute to drug insensitivity, drug resistance, and also resistance to immune therapies. And then we are really trying very hard to see if we can understand the early events of progression by monitoring circulating tumor cells. And so we are collecting also blood when the patients need clinical blood draws. We are evolving lots of collaborations, national and also international. We have several ongoing collaborations already. We are developing new ones. One of the tools that we use is a material transfer agreement in order to receive material from collaborating institutions. It is a collaboration. We want to enhance the circle because this is a disease that occurs everywhere, and but at any one place, not a lot of patients are being seen, and so we need lots and lots of different inputs. Sorry about that. This is a little bit jumpy today. So I'll tell you a little bit about Project One. These are all the people that involved in Project One. This is just some of the people, actually. People like Li Wei Bao. She's been in my lab for 26 years. And she's going to be at the front line. If a sample comes in and is delivered at 8 in the morning, she'll be the first person to open the box and begin working on the sample that comes in. It doesn't matter what time of day or night. These two individuals will take the sample and begin preparing the DNA and the RNA of that sample to be studied in depth in sequencing and multiple other ways in which we analyze the samples. This individual, Aaron Udager, is a pathologist, so he'll be overseeing all the sequencing, the proteomics. And this man here, Nate Merrill, is the person who really developed the whole platform that I will show you in a second. And Dr. Chin already showed you some data from that platform. Laura Gu is implementing a lot of the consenting of the patients, directing a mini clinical team that does the consents. Dr. Kiran Lagisset is studying the immune system. And Xu Cheng, there isn't an experiment in mice that he cannot do. So again, he's somebody who comes in at 3 in the morning to get the experiments done on a regular basis. So the Project One objectives are to test minimally cultured cancer cells from patients. We want to preserve the tumor heterogeneity. We want to come as close to understanding exactly the live cells that are causing disease and symptoms and affecting the quality and length of life for our patients. So those cells coming out of the body, whether it be a biopsy or surgery or circulating tumor cells, we want to understand the live cells. We don't want to freeze all the cells and then look at the drug sensitivity. For some experiments, we can freeze cells for sure. And Project 2 experiments that Dr. Su and Dr. Spence are doing, you definitely can freeze them. But for the experiments that Nate Merrill is leading on the platform of drug sensitivity, if you were the patient, you would want your live cells just taken out, tested, not some other derivative, not something that was banked and then resuscitated 3 months later. We have tons of data, over 7 years of developing this platform on multiple cancers, that the drug sensitivity changes. So that will be unacceptable to use methods that do not preserve as close as possible what's going on in the patient. We need to molecularly characterize the patient-derived organoids for sure. We think that the molecular characterization is critical to understanding the processes and why are some cells more likely to be killed by certain drugs and not others. Excuse me. Right now, what's happening with the tumor is there is some sort of a profile done, not always, but usually some sort of a profile done, which is usually a commercial, excuse me, a commercial profile. That is a panel. It's called NGS, but most of the time it's not whole exome, although sometimes it may be. And then there is some unranked list of therapies. Now, this is in general. As Dr. Chen said, we don't have anything beyond third-line, and lorlatinib is the only approved third-line therapy. So it isn't that there isn't a lot of other drugs just specifically for ALK that we know are right now available. We are repurposing some drugs, and others hopefully will be approved in the near future, and definitely we want to contribute to that. What we do, however, is a lot more extensive. Our molecular characterization is orders of magnitude more extensive than the commercial clinical characterization. We not only look at mutations, we look at copy number, we look at gene expression, we look at DNA methylation, we look at microRNAs, we look at reverse phosphoprotein arrays. That means we are interested in the proteins, but not just the amount of protein, but how much of the protein is active. Many of you know all about kinases, and you know that the kinase has to be phosphorylated or dephosphorylated in some cases to be active. So all that plus the clinical data. So what we want is to evolve from this type of just an unranked list. What we are hoping to do is to have a suggested ranked therapy for each patient that we test, with extensive sensitivity and resistance information, so that the patient and the clinician can make an informed decision. So if a scheduled biopsy is going on, we are able to bring tumor, the patient tumor is resected or partially resected. We are able to dissociate all the cells. We don't select the cancer cells. We plate all the cells. We just dissociate it because they need to be dislodged a little bit from connective fibers and other tissue that is not, that will not allow the cells to manifest themselves in the dish, and then we plate them. So these organoids that are growing here, that you see here, contain also immune cells that came with the patient, and other cells, endothelial cells, fibroblasts, other cells that are around the tumor. We are able to, in an automated way, test multiple drugs. So this is the organoid. If we put the drug on, the organoid dissipates, disintegrates when the drug is active. Here you have two drugs. This one, the red one, is more active than the blue one. We have at the same time the clinical history superimposed. And if there is enough material, we are able to develop new animal models in perpetuity so that they can contribute to research for years and decades to come. And we have been successful in doing this, both with material from pleural effusions, from surgeries, and recently a big breakthrough from CTCs as well. The advantage, of course, is then we have a surrogate. We have the mouse as a surrogate for the patient, and we can even test whether the drug therapies that we propose are working or not. I want to point out something that may be a little too technical, but I don't believe it is because it's so important. In our work, when we say that a tumor is sensitive to a drug, what we mean is that that patient could be treated with that drug at doses that are approved for that drug and that disease. This is a critical thing. A lot of drug testing happens at 10 micromolar. Unfortunately, many drugs cannot be given at 10 micromolar. So the fact that they kill some of the cancer at 10 micromolar in the dish It's nice to know, but it does not help the patient today. We, on the other hand, for the 1,500 or so drugs that we have available at our disposal, and we can choose any combination of those, any subset of those, depending what Dr. Chin thinks we should be testing, depending on the tumor. We definitely will test all the ALK drugs, etc. But many times we don't know if somebody has ALK. We still collect the sample, and then therefore we do a larger panel. We will always test the Cmax. That if we have to pick a one concentration to test, we'll always test at the known literature or published concentration or known FDA approved concentration for that drug. And this is a really critical, important thing. And so this is doable in the patient. So I'll show you some of the data, and then we can stop and answer questions, because I think a lot of people are very interested in that. Now, this is just to show you what we call, the spaghetti curves, right? These are sensitivity curves. This is how many cells are viable as a function of the concentration of the drug. This is very low concentration because this is a log scale and this is -10. So tiny, tiny, tiny amount of drug. It doesn't kill anything, right? 100% of the cells are still there. As you increase the concentration, some drugs don't kill anything until it's much higher. So that drug is not very good. Well, that particular drug here is Incertinib. But this drug here is ALKtALK, not very good either in this particular case. So unfortunately, we find that a lot of the drugs that the patient have been taken, of course, they have progressed on. So how robust is our platform to reproduce what's happened to the patient before is nearly 100%. If somebody is resistant to ALKtALK, we'll find them to be resistant to ALKtALK. And this is very important because it's important to have a positive predictive value for sure. We want to be able to suggest drugs that are going to work on the patient will do well. But equally well, if the platform is robust, at the very least, it should replicate what has happened to a patient, which we already know has happened. And so that's super, super important. And we've been able already with the patients we have had to enhance our negative predictive value together with other tumor types that we've done. And so in general, the tumors are highly resistant by the time we have them, because of course patients have progressed. Although we are often able to see something that the patients are maybe a little bit more sensitive to. I wanted to show you something real quick, and that is you will hear a lot about IC50, which is the concentration of drug that kills 50% of the cells. Now, if I'm a cancer patient, and I am a cancer patient, I want 100% kill instead of 50% kill. So I want a drug that's really effective in the sense that I don't have to take a lot of the drug to kill my cancer cells. But boy, if I have two drugs to choose from, I'd rather have the one that kills all of my cancer cells, not 50% of my cancer cells. So here is an example. Here is lorlatinib. This patient is already resistant to lorlatinib, but even under the best circumstances, even if you are able to give this much lorlatinib, this curve plateaus at a little bit under 50%. So, and that was probably true. I hate to give this news, but that was probably true from the beginning. There was probably already, by the time the patient presented with metastatic disease or advanced disease, there was probably already a clone that did not respond. And therein is the importance of doing this work as often and as thoroughly as we can. So here you see that even though this tumor is highly, highly resistant, there are some options for this patient. Whereas these are the drugs that they got before, and the red means insensitive. I want to show you another example. Here is a patient that was undiagnosed at the time of resection. This is a patient who had no drug exposure. Now, I just showed you before what a resistant curve to alectinib looks like, or to lorlatinib, right? So here it is. Now, this is exactly what I just told you. Even though this tumor was small, it is small because it looks small compared to the size of the lung on the CAT scan. When the tumor hears us say a 2-centimeter tumor is small, it's laughing at us because a 2-centimeter tumor has close to 2 billion cells. It's not small from the tumor's perspective. And here is the proof of what I just told you. This patient's tumor has never seen alectinib. This tumor is already resistant to alectinib. Even if you could deliver this dose, the most you would kill is about 70% of the cells. This tumor is only 2 centimeters. So this tells you how much we need to understand. Right here is the key to how much we need to understand how this tumor advances and progresses even through the early stages. And so here you see profound resistance to lorlatinib, crizotinib, alectinib, but some sensitivity to several other potential options. So it isn't that they're resistant to everything, but unless you test it, you wouldn't necessarily know. And of course, these drugs are not all the same, and they would have a different spectrum of side effects and whatnot. I just want to show you how much the patients have contributed to our understanding, even in this short period of time that it's been our honor to interact with these amazing patients, amazing families. And if any of you are in the audience, you have our eternal gratitude, and I hope the eternal gratitude of the cohort of patients, because without you, we can make no progress whatsoever. Now, this is a case where we received both a pleural effusion and abdominal pelvic effusion, a paracentesis and a thoracentesis. The patient underwent both withdrawals of fluid. And this is an incredibly rare experiment to do. And it really proves how robust our platform is, that you have a very hard time seeing any difference. But these are entirely different parts of the body, entirely different samples. It's the same patient, but it should be the same, how it looks the same. But there are so many variables involved in testing all these drugs, what cells are coming from in each sample, all the differences. But the meticulous treatment of these samples that we undertake is really, really extreme. We put everything into it. We cut no corners. We are pouring decades of understanding in my laboratory of how cancer cells behave and how metastases arise to this problem. So in the interest of time and being able to answer questions, I'd like to stop here, and hopefully I give you a flavor of what we are doing and we can answer some questions.
Terrific. Thank you very much. I learned more in half an hour than I have in the last 2 months. Okay, so let me see how we're doing. So
I see 32 new messages.
yeah, there's a lot of them here.
Okay.
I've been just answering the questions as they come.
I know,
Oh,
and I've actually been asked to repeat them because the vast majority of the people that will see these videos will be seeing them on YouTube. So we're going to walk through a few of them, the best ones, if we can. Is that all right? Angel?
Yeah, absolutely.
Do you have any hypothesis for how Gilteritinib may differ in terms of treatment from fourth generation ALK TKIs?
Yeah, so I think that was a really great question. So TKIs, our ALK direct specific TKIs are still specific to ALK. And so fourth generation ALK TKIs are just designed in a way that they can bind to the binding pocket of ALK in a different way. But Gilteritinib is not only inhibitor of ALK, it actually also has activity against these other kinase domains. So it's a lot more broad. And so the theory is that fourth generation ALK TKIs like NVL-655 would be effective against patients who still retain an ALK mutation at progression, whereas Gilteritinib may have effect against both ALK mutations and potentially non-ALK mutations because it interacts in a different way.
So I had a couple of my own questions on Gilteritinib. How's the toxicity profile?
That's a great question. So from the clinical trials that have been done that are in leukemia, understandably a very different disease process, it seems to be generally fairly well tolerated. So we do watch out for cytopenias, though that may be a bigger issue because these are patients with leukemia and that's a marker of their disease state is that they have fragile bone marrows. We see, of course, some fatigue, some GI side effects. I think somebody had asked about potential pneumonitis that had not been seen. There is something called differentiation syndrome, which can happen, but again, that's a leukemia-specific phenomenon, not expected to be seen in ALK non-small cell lung cancers. I spoke to my colleagues in leukemia who use this drug, and that is their impression is that generally is quite well tolerated.
And is it a long-term course of treatment like the TKIs, or do you do it as?
It's going to be a daily continuous treatment.
And do you stay on your TKI as well?
No, that has been brought up to consider combining Gilteritinib with another ALK TKI like alectinib or lorlatinib. I think this study hopefully will open the door to many different explorations of how to best use this drug. But I think when we don't even know how this drug alone would work in non-small cell lung cancer, we didn't want to potentially create more toxicities or muddle the picture by adding it on with another ALK TKI.
Got it. Thank you. Looking through.
As Jeff is looking through some of the questions, can you walk us through how to become involved in this study? How do we go through the process of, giving our tissue?
So Jeff just had this amazingly informative piece that really describes the study. We are delighted to hear from patients, and I should put the.
I just put up the
you put up email. Okay, yeah. So I think we are delighted to hear from patients. If you are and you're probably outside of the University of Michigan, we will definitely run it by the clinical team. Your query, either Dr. Chin or one of our clinical team members will answer you. Would like to engage you in identifying your champion doctor at home in your at the place you're being treated, or you are going to undergo a procedure. Sometimes it's a different place. You may be treated with an oncologist in private practice in your area, but you're having a procedure at a hospital, regional or tertiary care hospital, whatever it may be. So we will need somebody at the hospital where the tissue or the blood or the paracentesis or thoracentesis will be done. And therefore, that the sooner that happens, the better. You do not have to be having surgery tomorrow or next month to access us. In fact, we like to hear from you when nothing is happening, and you're doing very well, because then we have more time, we have the luxury of time, but you are interested in being part of the study. We can sign you up, we can talk to you about the consent. If you're interested, after we discuss the consent, then we'll sign you up, and we begin the process of having a material transfer agreement, and so that at the very least we can begin to have your bloods when you get your blood drawn, for your doctor to see how your liver functions are doing, or whatever reason you and your doctor choose to have your blood drawn. It'll be 5 tubes of blood. It isn't excessive. You will not become anemic from it. And it will allow us to harvest these circulating tumor cells and study them in depth. As I said, we've even succeeded in making an animal model already, and we are trying for more. We find that ALK patients have a lot of circulating tumor cells. We are somewhat surprised by that, in the sense that we expect it to be something, and it's about 2 or 3 times higher than what we expected based on other epithelial cancers. There hadn't been extensive studies, so we didn't really know exactly what to expect.
So it's an aspiration for you guys to provide analytical feedback as to a person's cancer down the road. And that relates in turn to being CLIA certified. Could you discuss that a little bit, where you're going down the road?
Well, it's very good news in the sense that I think we are ready with the help of all the patients, their continued help. We still need more experience with ALK per se, and with lung cancer in general. But other kinds of lung cancer are obviously quite a bit more common, and so we still gather a lot of useful experience, but we want more ALK because ALK is our focus. So I have identified somebody who's coming to work with us in the next month or so, and she will focus on getting us CLIA approved. So we have the lab space. We will have to create the whole drug testing facility. We'll have to replicate it for CLIA approval patient material. So we have to basically mirror the lab, the research lab completely. You can't do research and be CLIA in the same equipment. So that's going to be a huge undertaking, but we can do it. I mean, it's just, we know how to do it. So we'll create a new lab. We have, as I said, identified the space, which is incredibly short supply at the University of Michigan. It's one of the hardest things. So I am very hopeful that in the course of this year, it will be CLIA approved. I'm very hopeful.
And what is your aspiration for providing information back to the oncologists of the patients that you're studying?
Right. Well, right now we are not allowed to give results. The patients sign on that they agree to not receive results. That's just what happens, because you either give results officially or you don't give results at all. There is nothing in between that is recognized. But I just show you data. That's data from real people and real patients. And so on an individual basis, we definitely can give an update to the collaborating doctors who are our scientific partners. They are invited to see data, to opine on the data. They are our scientific collaborators, and that's completely allowed. They are not, this is not a report. This is not a clinical report. And it is not per se actionable then. Eventually, we hope it will be, and that's where we are heading. But in the short period of time that we've had, in spite of tremendous initiative on the part of the patients, and again, I'm incredibly grateful and humbled by everything the patients do, and the families is incredible. is just unbelievably incredible. We often fail to collect the samples because we can't process the MTA fast enough. And obviously, we are number 1, number 2, number 3, number 20. The only thing that matters to us is the patient be well and get the best care. So that's more important than anything. And so that always takes precedence. And so the more we can talk to each other and participate when the situation is cool and not much is going on, the easier it will be to make progress, I think.
So
So
patient. Oh, sorry, Jeff.
I would encourage all of you to jump on the Facebook site, the ALK Positive Facebook site, and look up my recent write-up on the University of Michigan's program. It lays out the whole program in some great detail with the contact information as well. And it gives you a kind of roadmap for how to collaborate and how to participate. And I want to make a point that's important to the people at University of Michigan. This is a medical collaboration between you and your oncologist and your treating clinic or hospital. And it's a long-term relationship that provides a lot of information spread out over a timeline so that you can be watched and the progression of your disease or the lack of progression can be watched. So it's important to create that relationship by getting in the process. And that process will be different for every client. But again, if you look under Jeff Sturm or you look under University of Michigan on the Facebook site, you'll find a real detailed write-up that'll answer a whole lot of questions. One question I've been getting repeatedly is, can patients from outside the country, outside the US, participate?
Yes, they can. They can, absolutely. Again, we need to meet, we need to have a Zoom call or something, meet with the oncologist or the head of the clinic or whoever it is, and see if they are interested in being collaborators. We just had a very productive meeting with a Finnish group who have archival tissue. Even if there is not a particular patient, we still want to sign an MTA with that group because they have a pretty good archive. And we also need archival tissue, that is historic tissue, to understand a lot of our hypotheses that come from the Project 1, 2, and 3 work. And so there is a lot of interest in international collaborations. We are meeting with people from Israel, UK, Sweden, Denmark. There's an ALK Positive in Denmark now that we gave a webinar to as well, and so on. So we are hoping there will be more collaboration.
So there are a couple of questions on the material transfer agreement, which you've answered, Angel, somewhat. Can you just tell us a little bit about how successful patients navigate their own clinics with the agreement?
Yeah, you go ahead. Angel.
Oh, yeah, no, I think the most important thing is to have a sponsor at their own institution. Usually it's their oncologist. We recently had a patient whose sponsor was their surgeon, could be their medical oncologist, but really a doctor that they're working with that is interested in this collaborative process, because it's really, Stacy in our group is really great. She has the MTA that is written up. It really has to go through the other institutions, regulatory committees. And so that's why having that champion for you at your own institution is so helpful. There's obviously red tape that we all try to get through as fast as we can. We often encourage you to connect us with your sponsor ASAP. So tell us who it is that you would want us to talk with at your institution to get the MTA going. So Dr. Mariah mentioned, it doesn't have to be at the time of progression. We would prefer not to be the time of progression, because then we're working on a deadline And we don't want to delay anyone's care. If you are like, I think down the road, I would love to get on this research initiative. I'm doing great on elective lorlatinib. That's fantastic. We would still love to have you join us.
And that's a critical point. You don't have to have the tissue to sign up for the study.
Right.
And you may not even, am I correct? You may not even donate blood at the initial.
Correct. You can simply just sign up with your name. And just, we have lots of patients who just kind of want to be in our database, so to speak. We just send you a simple survey that just asks you to give us a little bit of a story about your journey with ALK thus far.
Yeah, if I could say a couple of things. I mean, it is so important for patients to understand this. This is, I know we are blazing the trail, but in general, patients do not understand necessarily their rights. When you go to have surgery, you basically are signing away your rights to excess tissue. If you look at your informed consent for surgery, in most institutions, you are relinquishing the custody of your sample. Now, it is very difficult to not do that, right? You want your surgery, you want your procedure. The last thing you want to do is offend or antagonize your doctor. Absolutely, I don't recommend that. But from an ethical point of view, this is your tissue, and you are voluntarily relinquishing control. So the material transfer agreement would not be needed if you were in control, because then you would say, here is the box. I want you to put excess tissue in this box according to these instructions, and I will mail it. But you have relinquished that, and also you are asleep and anesthetized, and But I think it's important for patients to understand the origin of the material transfer agreement. It isn't arbitrary. It is that you, the patient, I relinquished my control when I had my surgery for cancer, and I did it quite happily. And hopefully somebody is studying my tissues. Most likely not, but I would hope so. But I can promise you one thing: not one cell that comes to our initiative is not immediately studied. We do not bank things to decide later what we want to do. We 24/7 look at every sample. And talking about international samples, we made it on the wire from British Columbia. And a week and a half ago, we had a fantastic sample come in from British Columbia, including the doctor rescheduled the patient by a couple of days. It was completely safe for the patient for that to happen, so that we could get the sample. I mean, God, that was such an honor to have this happen, but it can happen. We have a very standard pared down material transfer agreement. And so we make it as easy as possible. So yeah, I'm sorry that the patients have to learn about one more thing, the material transfer agreement, but the origin of it, I wanted to explain, is because we relinquish control of our sample.
I've read it, and it's simple. It really is easy. It's finding the right sponsor within your hospital or clinic that can walk it through their legal department, which, not everyone is skilled at those kinds of things. But I think it's important to note that you're going to need to hand your oncologist something. Describing this program off the top of your head as a patient, I wouldn't even try to do it. So the write-up that I've done that's on Facebook was intended to be not only educational for the patient group, but a handout for your oncologist. And I believe the University of Michigan group is preparing additional collateral material to be able to hand out for the patients to be able to hand out to their oncologists. That's under review right now?
Yes, it's under review by the IRB. We finished editing and yeah, I mean, we are a little behind the 8 ball on that. I totally get it, but there's been a lot to do. And, we need better patient material. We need to put your article on our website, Jeff. And there are a few things that we need to do. Yeah, you just did an amazing job. And
Thank you.
yeah, in spite of
While I
us
was-
giving
while I
you
was-
an enormously hard time about.
Yeah, exactly. While I was in bed with COVID, by the way, I might add.
I know Lorlatinib, like you say, right?
And on Lorlatinib, exactly. So in order to wrap it up, we want to be respectful of your time. Could you define equity of access? What does that mean?
Well, this means what we just discussed. We want to empower every patient. This costs nothing. We pay for everything, from the shipping, for everything. We want every patient to be contributed to their tissues. If you don't contribute your tissues, your genes are not represented. What happened to you in life is not represented. Two people have the same mutation they are born with, say they have a mutation BRCA1 gene. One patient will get cancer at age 30, the other one will die at age 92 of an unrelated problem. How did that happen? Your trajectory is only your own. What happened to you growing up? What were your exposures? What did you eat? How did you suffer? Did you worry about safety? Did you worry about housing? Were you evicted? Were you constantly subject to stress? That causes all kinds of inflammatory problems, all kinds of procarcinogenic events. So everyone's trajectory needs to be represented. That's what I mean by equity. Because unless you are represented, we don't learn about you. And that's what's happened historically in oncology. At least we haven't been as bad as cardiovascular disease, where basically for many decades only did research on men and middle-aged men at that. But then all of a sudden we discovered that women are dying of heart attacks too. So whoops, let's include women. We are not as bad as that in oncology, but because we learned, they blazed the trail of understanding even gender representation, but we are so far behind equity. And there are such disparities of outcomes. I mean, I can't believe we are reading in the newspaper about maternal mortality in downtown Detroit, right? It is worse than in some sub-Saharan countries in Africa, where I have worked in the past, trying to improve maternal mortality and cancer care of young women who were pregnant with cancer. So, it's just so much to do. I want all the patients who are on this call and to talk to other patients that to feel empowered to participate in research. This is research for the good of all. And it is super important to us that it be done that way.
Angel, any further comments before we sign off?
No, I think, like I said, I've been trying to answer the questions that are coming through. So hopefully people have found those helpful, but really just please encourage everyone who's interested about any part of our research just to email us.
And
Somebody's asked one question for the MTA. Don't these tissue samples need to be portioned out between cancer centers? So if you sign more than one consent, then whatever happens at surgery, there is enough. It'll go to whatever consent you sign. But occasionally, there will be more than one research study. If you decided to sign for more than one research study, if you have a very small biopsy, you may want to decide which study you prefer. I'm not saying you should choose ours. I'm just saying you should decide, because it's unlikely that there'll be enough for more than one study, is my guess. But, it will vary from person to person. Thank you.
We good?
We're
Thank
good.
you.
Thank you so
Thank you
much
all.
for joining us. This has been so wonderful, and we would love to have you back again for an update soon, or at least to discuss the Gilteritinib trial as that gets moving along. So this was wonderful, and then we'll get this processed as soon as possible and out on YouTube. And there were so many great questions in the chat too. I'll see if I can play with them and make them private, or so there's not any names shown, and get that out there. But thank you both so much for all of your time and for
Thank
joining.
you.
You guys like Jeff, come back soon. He'll be speaking. Was it April 2nd?
Oh, yes, April
Yeah,
2nd.
April 2nd will be our next ALK Talk with Jeff, running part 2 of our ALK series.
I have my very own hour, and it'll be for the first time in ALK Talks, and it'll be all about assembling and managing your care team. And I think it's going to be quite interesting. We'll tell some stories from my wife's breast cancer and my 2 cancers, and how we've navigated successfully for the last 15 years with it all. So look forward to seeing you.
Absolutely.
Thank you, Dr. McIver and Dr. Chen. Really appreciate it. And join us back, and we'll see you soon.
See you soon.
Thank you
Good
all.
life.
Thank you.
Thank you so
Bye.
much.
Thanks for all you do.
Thank
Thank
you so
you.
much.
Bye.
Thank you.
Bye.
Judith.
Thank you.
And thank you, Judith. Yes.